So technological strategies to improve safety of fresh food is the topic today. And I've chosen fresh food because that is the trend of the consumer to eat more fresh food. But once you start changing your eating habits, then you also have a range of food safety issues that you need to address. And I think we are at a stage that the consumer needs and the food industry they're trying to match them together. But there are a lot of food safety issues that we need to address in the process. And any technologies that are being developed to address these is some of the other things that I'll be touching on. So I'm not based at St. Lucia. I'm based at Cooper's Plains, and it's called the Food and Health Sciences Precinct. I'm truly blessed to work in laboratory conditions here because it's state-of-the-art. And I always think it's like the hospital of the food, because you can do like clinical trials, light up to clinical trials. So if a food industry comes to us, we can do all the processing in that pilot plant for it to be sold in a market into the retail outlets if we can get the necessary aqueous approvals. So we really do, we engage with industry a lot, and we try to take research to market. So for us, that is a big priority within the research group that we work in. And Heather, Mary, all of us sit at Cooper's Plains. The facility is shared by a lot of scientists, CSIRO and Queensland Health, QAAFI, and it's owned by Department of Agriculture, Fisheries and Forestry. So a lot of our projects are in collaboration with scientists in DAF. So an overview of the presentation would be what is the food safety situation in Australia and globally, the incidence of food poisoning, what are the challenges for food safety with the new consumer trends and technological strategies to improve safety of fresh food, and some commercial successes that we have had while working in this area. So for those who don't know this, I'm just giving an overview because Sheila is here as well, so maybe she will understand a little bit about what the Australian food regulatory system is. Sheila is one of our visiting scientists from Kenya, and we are very happy to host her. So if you look at the regulatory situation, Australian states and territories, they would look after public health and food law, which looks at the safety of food. And Commonwealth government would look at the Food Standards Code, which is the Food Science Australia and New Zealand Code. So that's where all your the levels of additives and what is allowed and what is not allowed is given in the standards. And then the enforcement is where aqueous would come in for imported food. And then local government would have their own enforcement regulations. So because we are in Queensland, what Queensland does is we have the primary production, which is governed by the Department of Primary Industries and Fisheries Stock Act of 1915. And then Safe Food Production Queensland is important to us because I'm in the processing area, and they look after all the processing and manufacturing. And then if there is ever an incident of food poisoning or issues, Queensland Health would act under Food Act 2006, and they test a lot of samples to identify what the issue is. So that's the overall QLD structure for food safety. Now, if you look at the latest food poisoning issues that have occurred, it's not only in Australia, it is a global problem. In British Columbia, Canada, they had cheese that was contaminated with E. coli because of using raw milk, and it was the Gouda cheese. In Australia, they had problems with Campylobacter in the poultry liver because it was undercooked, and people felt very ill after eating the poultry dish. In Colorado, USA, there was an listeria outbreak in cantaloupe melons, where washing the melon surface is a little bit of an issue because of the rough surface and striation. So you can get listeria going into the crevices, and it's very difficult to get it out. I think about 33 people died, and these farmers had to, they were taken to courts, and then they were charged. And you'll all remember the bean sprouts E. coli 0157 outbreak that occurred in Germany due to the bean sprouts, and 22 people died from that. So this is kind of a global scenario. We want to eat healthy, but with that we also have issues of other microorganisms coming up, emerging pathogens coming up, the virulency rates going up. So we have to address all that. So according to OzFoodNet estimated, this is like the Center for Disease Control in America. This is the one that you have in Australia. They say about 5.4 million cases of foodborne diseases per year, costing about $1.2 billion annually. And 100 outbreaks, and a lot of it is related to fresh produce, and internationally distributed food is increasing. So due to these reasons, we have to address the issue of how do we keep food fresh and safe. So the food poisoning incidences are on the increase, as I told you before, because of the changing eating habits of people. They want to live a healthy lifestyle. Food production methods. So we have very large companies that have taken over smaller companies and producing food on a global scale. So if there's one incident of food poisoning, it'll spread very widely, very fast, because of the globalized food supply chain, and also the change in demographics. So, working single mothers, both parents working, we're looking for convenient foods. An aging population, they will need other types of food, or people who are very sick. So there's a lot of change in demographics that we have to address. Now the consumer is also having their issues of, we don't want too many additives in food. We want less chemicals. If I don't know whether you've read the book In Defense of Food by Michael Pollan. He's a food critic, but he said if you walk down the supermarket aisles and if your grandmother doesn't recognize the food, then don't buy it, because it's so processed, you don't know what you're buying. That was the indication that he was giving. So people want to recognize the food that they buy. They don't want it in a form that they don't understand. So when you process, you do add a lot of chemicals and additives, and they don't want a lot of E numbers. They want clean green labels. And then there are also mixed messages about the dangers of additives. So Aldi would be a very good example, because they had that issue of food coloring and chemical preservatives. And they said that children were going to be hyperactive. This was a study that came out of England. And Aldi, on its own, said we are not going to have any artificial colors. We'll go for natural colors. And now they are moving towards natural preservatives as well. So this was a retail chain that on its own decided, because the consumer wanted natural and not any artificial preservatives or colors, they decided to remove it. So they've lost faith. They're concerned about preserved food, and their desire is for natural and non-processed fresh food. So what are the technologies that are available to address some of this, keeping the food safe and then keeping it for a longer time and keeping it fresh? So packaging is going to play a big role in the future towards achieving this goal. High pressure processing is quite promising, a little expensive at the moment, but still, because there is no cooking, you still get the flavors and all those notes preserved. Modified atmosphere. Again, you can work with fresh foods with modified atmosphere. We are looking at light treatment, which is, we call it photosensitization. It's a unique technique that's used in the medical field, but we think there'll be a lot of applications in the food area. And natural antimicrobial. So we are looking whether things like plant extracts can actually preserve food and remove the chemical preservatives like the metabisulfites and the benzoates and the butyl hydroxyanissols and all these artificial antioxidants. So my area in the past 5 years, one of the areas that I work quite intensively is looking at plant extracts for shelf life extension. And it's very exciting, because they're synergistic compounds together. They're phytochemicals, and they can give a lot of positive effects on a food system. So they can have an antioxidant property. So if you have antioxidant properties, then you can retain the freshness, because it will absorb the rancid off notes that are coming out of a food. Antimicrobial, so it can preserve the food by inhibiting the formation of the microbes or playing around with the communities of microorganisms that are present. Anti-browning, if you, things like avocado and apples or some of these products, it's an issue, so you can use plant extracts. So it's kind of a lot of positives when you use plant extracts because with one mix, you can have a lot of hits. So plant extracts alone does not do the trick. We need to use other technologies, and we use hurdle technology, so that will be the packaging or high pressure processing to give that added shelf life. So we've looked at, we find that native foods are very exciting because the phytochemical composition is very high and it's very potent. We worked with 2 plums, Davidson plum and Kakadu plum. And if you look at the petri dishes down there, Davidson plum is as effective as the oxytetracycline, which is the antibiotic, against the Staphylococcus aureus, which is a food poisoning organism. And there are different phytochemicals there, like the anthocyanins, like the gallic acid, ellagic acid in the Kakadu plum, and it also has other acids like the ascorbic acid and the malic acid. So they all work in synergies. So in terms of building up resistance, it's very difficult for the bug because the bug gets hit with different chemicals. So a lot of the antibiotics, you find that a pure chemical will be affecting one bug and then it creates a resistance. But in this case, you get like a whole range of molecules that will affect it. So it's advantageous when you work with the whole ingredient because you also can have that clean green label. So you can say herbs and fruit extracts or something like that without having E numbers, because this is actually directly from a plant source that people have been eating for 40,000 years. So it is still food that we add as a functional ingredient to another food product. So this cross-industry application of native foods into other foods have actually benefited the native food industry because it is an emerging industry. It is small, but I think there's a lot of promise. And then if you apply to other industries like the meat or the seafood or horticulture, then you open a big market. And then native foods are either wild harvested or they are grown as orchards. And if you look at the wild harvest, then you can have economic flowbacks to the indigenous communities who work in gathering these native foods. So it kind of is very exciting when you look at it that you can have all these flowbacks back to a community that does that as a natural thing. So the seafood industry approached us. This was done in collaboration with the Department of Agriculture, Fisheries and Forestry. We have a very strong seafood team. And they wanted the shelf life of cooked chilled prawns extended to 14 days and the retention of the quality. They wanted metabisulfides out. So they were really addressing that consumer need for removing chemical preservatives or additives. So because we already had a project on native foods when we were looking at natural antimicrobial properties, and they also had a criteria, they did not want the flavor of the prawn changed under any circumstance. So if you have smelled some native foods like lemon myrtle, anise myrtle and all, you get a very strong smell. So you can't put that on a prawn. People will, the prawn industry wouldn't like it. So you had to go with something that would have a bland aroma, but still have that efficacy. So our approach when we do get a problem like that is first yourself to understand what the microorganisms are within that food system. And if you can actually inhibit the spoilage organisms, you kind of also have success with the pathogens, because the spoilage organisms are like things like pseudomonas, which are gram-negative, are quite difficult to inhibit. So we isolated all the organisms that are present in the prawn. And then we screened a lot of natural antimicrobials against the specific spoilage organisms. And then we looked at a range of packaging options that we could bring together for the chilled prawns. And then we trialed with antimicrobial solutions and packaging options. So we went through really bad smells. So we had people sitting around and smelling these test tubes that were really horrible. But we finally, I think, successfully managed to increase the shelf life. So when you're working with industry, another thing is that they don't want to change the process that they're following. So you should try to put your solutions into already a process that's available. So we thought the dip would be ideal to put it into the chilled solution when the prawns were cooked and when the temperature was dropped. So that's where we thought it would be ideal to have a solution. And then, of course, draining and storage and packaging is something that the industry already does. So the only additional step was adding this solution to that cold storage, I mean the cooling tank. So you can see that is the control sample, which was the blue line, and we got about a 3-log drop at 14 days. But you always have an abuse period when you do shelf life studies because you want to, because the distribution chain would give some stress to the product, so you always want to get 4 or 5 days in excess of 14 days that you're sure that it lasts for 14 days. But then what does this mean? So we went on to look at bacterial community profiling by pyrosequencing just to understand what happens to the the other communities with all these spoilage organisms. So we have Sorab here from AGRF who did the rest of the analysis for us when we extracted the DNA and sent it to them. But what they did was pyrosequencing, and you can study microbial communities in any environment. It can be a wound, it can be a sick animal, or a bacterial biofilm. So in our case, it was the prawn itself. And then you amplify the PCR, and then you get the amplicons, and then you fragment it, you attach it onto beads, and then you put it on the picotiter plate, and then you get a phylogenetic identification. And AGR gave us the possible phyla that can be present. And this is what we did. So we went to a prawn farm, we took a control sample, and we did a treatment. So it's, when we say cacodoplam, it's not only cacodoplam. The formulation is much more complex, but I did use a native plant extract, which is cacodoplam, which is bland. It doesn't have any aroma or flavor, but very high in ellagic acid and vitamin C, one of the highest in the world. And we sampled from 2 ponds, and we did the conventional microbiological analysis and the community profiling. And you can see at 14 days, we still maintain that 2 to 3 log drop for the standard plate count. And if you look at the change in the community, so you got different communities like the planktomycetes and the actinobacteria. But what is of interest to us is the proteobacteria, which is the Shevonella and Pseudomonas, which are the spoilage organisms. And you can see for the control at the end of 21 days, the microbial flora had completely changed to the proteobacteria. Whereas if you look at the treated sample, that change was much less. So you did get Shevonella and Pseudomonas form, but at 14 days it was much less than the control sample. So indicating that it is inhibiting the spoilage organisms, and that's why you didn't get all those amine notes and all those sulfur notes that you get in a spoiled prawn. And then if you went deeper into the phyla of proteobacteria, you found that the Pseudoaltermonas, that family was very high in the control. Again, you get all those spoilage notes and off flavors happening. But in the Cacodus formulation, you got much less of it, and that had a significant effect on acceptability and sensory qualities of the end product. So it's interesting to understand that the microbial communities, what the microflora is, really balances the formation of these spoilage organisms. So this was quite a successful project because last year we had almost 500 tons of prawns being dipped in this formulation, and that is like we made all the solutions for the industry, and almost all the prawns in Queensland were dipped in this solution, and a lot of your Woolies outlets would have had this solution. So all that was prepared in our labs with the DAF staff. These plant extracts can be done with various food systems. So this was just an example where we looked at papaya. This was funded by the Queensland government, again in collaboration with DAF. They wanted to use papaya as a fresh cut product. And we looked at limonene, cinnamon, aldehyde, and vanillin as the phytochemicals that we were going to add. And you have to have very clean conditions. So if you start with high microbial loads, whatever you do, the product goes bad. So you have to have very good hygienic conditions when you process fresh produce. And this is something that you really want to see. So the vanillin, you can see, even at 18 days, it is going down the counts, and that is very good. So if you get something like that, you have that abuse time, and still the counts are very low. So that is pretty good in comparison to the control. So there's like a 4-log drop. So this is what we would recommend to the industry, because some people don't like the flavor of papaya. You can add it, or you can have it in a beverage with vanillin. Vanillin is very good to extend the shelf life of even other herbal drinks. And you have the potential of developing a range of innovative fresh cut products with unique flavors from different plant extracts. So this is something that Mary and Sheila would be interested in. It's the Becca-Syro aflatoxin project, where they're looking at to see how aflatoxin can be reduced, or in cultivation what happens. My component that I'm interested in is looking at Aspergillus fungi and the aflatoxins at post-harvest stage. What can we do once it is harvested and once it comes out of the field? When these toxins are present, is there anything that you can do to decontaminate? So we come in at the post-harvest intervention stage, and we're looking at photosensitization as a potential decontamination tool to reduce the aflatoxin in maize. So this is used quite a lot in the medical area, and Ben, who's doing his PhD, is working on this area, and he's looking at photosensitization. So this is, for photosensitization, you need a photo dye, which is the photosensitizer, and you need oxygen. And you need light. So when light is shone onto the photo dye, which can be a plant extract, so we work with curcumin, we work with chlorophyll, you can have a natural plant dye. It absorbs the light and it goes into that excited state, and then it can give off its energy and become a triplet state, and then it can attack the cell and cause cell death. And you also get a lot of other, like hydrogen peroxide and the peroxy radicals being formed, which will damage the cell. Or molecular oxygen will absorb the energy and go to the single oxygen stage, which is quite active, and it will attack the microorganism and will cause cell death. Usually it's type 2 photosensitization that occurs. When you're working with microorganisms, fungi are a big problem. Gram-positive bacteria, I think, are easy compared to gram-negatives, but when you have funguses and spores, it becomes a big issue because they are quite difficult to inhibit. So this is just some basic results that Ben has had from curcumin as a photo dye, where you look at the maximum absorption of curcumin, and then you have the Aspergillus spores, and you bring the dye and the Aspergillus spores in contact, and then once the dye is attached to the surface of the spore and you shine light, it absorbs the energy, and then it kind of is inactivated, and the the spores are killed. So we've got 4-log drops, and GE is now currently working on chlorophyll, and we are getting some very good drops for Listeria monocytogenes. So, which is also which is a problematic pathogen in fresh food. So I think this technique has a lot of legs in the food industry if we can work with it, because if you work in the visible light area, it still can be looked at as a clean green technology. So curcumin was effective as a photodye and significantly reduced the spores of aflatoxin, and it can be we are trying to use it as a decontamination tool for maize in Africa, but it has other applications in other areas of food science. Okay, so this is the last part. I think packaging is going to be huge in the future, because of this issue of keeping food fresh. People want to go to the supermarket. They want to recognize that it's a prawn. When you have some green herbs on it, people feel very happy. It's really fresh and good and all that. So to do that, it's pretty difficult because every time you add a new additive or the herb, you have to all have microbial loads that are very low. So if you add pepper, pepper is a real problem. Pepper starts with 10 to the 9 colony forming units per gram. So when you add pepper, then you will have a big problem in your sauce or whatever. So you have to be very careful with what ingredients you select. And I think vacuum packaging works very well for this product because it clings onto the product. If you have a marinade or a sauce, it kind of very nicely gives the picture that it's in place. It's not all over the place and running all over the container. It doesn't have a lot of consumer appeal. But then also it removes the oxygen, and you prevent a range of microorganisms forming, and then also you can extend the shelf life. So it just depends on the food product and the commodity that you're working with. I like this one. It's called freezer to oven concept. It is it's not developed by us. We hope to be there one day, but not now. It's by Multivac and DuPont and Teijin in Japan. They have a patent on this, but this is a thermoformable films, so you can have this on chicken or pork or any fish, and you get the feeling, the consumer gets the feeling that you have prepared that product at home. So you buy this marinade, the chicken or pork crackling, and you put it in your oven, and it heats up. While it heats up and the temperature goes up, it's still in the packing material. And then at a point, the packing material opens, and then you get a kind of a drying stage. So you get the crackling pork coming out like crackling pork, and you get the chicken like a roasted chicken, and you get bread that can be baked. So all giving the concept that you have taken it home and you have prepared it, and that kind of sits well with the fresh, healthy eating lifestyle. So they're making lots of money on that product. You get all these products in the local supermarkets now. This is done by Tassel. And this is micro-perforated packaging, which is very good for extending the shelf life of fresh produce like the salads and the vegetables. So this was another project that we did in Harvey Bay with an indigenous community. So they were using warrigal greens as a leafy vegetable. They are used to eating that. And also we worked with sea celery. And Heather was on this project, and we extended the shelf life for 18 days. And now this project has moved to the Sherbrooke community, and they take products to the, I think, Rockley markets. So micro-perforated packaging was the one that really extended the shelf life there. So it's like the packaging has very small laser holes, and you get a modified atmosphere because of that passage of carbon dioxide and oxygen and water vapor that occurs during the storage time. So the challenge to the food industry and food scientists is to fulfill the needs of today's consumer in a globalized food chain. Technologies will have to be developed to ensure food safety in different food production scenarios. And I think it's exciting times if you are in this area, because the more challenging it is, you get a lot of satisfaction when you do have a solution. These are to all my colleagues who have worked with me on all the projects for the past 5 or 6 years. Thank you, and for all the funding bodies. Thank you.